Hello and welcome to this video on why cheese cultures use a mixture of bacteria and not just one. When you make yogurt, there's a greater reliance on something like lactobacillus bacteria. Sourdough bread uses yeast and lactobacillus. Cheese goes somewhat beyond this. There is no one true cheese culture. That is, the wide range of cheeses need somewhat different bacteria. That allows for the low fat but high protein and high fat but low protein and more, things like lactose in larger or lesser quantities. There is a wide range of other factors need to be accounted for. Some of the contents of milk leading to cheese are more or less important to some bacteria than they are to others. This is why you need a culture with different bacteria. Most cultures have several purposes, but generally you'll find they fall into four categories. The first being acidification of the cheese through lactic acid, development of flavors, texture, and then smell. The different results and substrates allow cheese to be broken down into different categories, or different styles as well. This is why we have so many different kinds of cheese. You may wonder why, for example, is Parmesan so much more dry and crumbly compared to a cheddar, a brie, or a camembert. The different available substrates really are dealt with by the use of two different groups of cultures, these being mesophilic and thermophilic. Mesophilic is a moderate temperature cheese. Fundamentally, you could compare it to something like an lager yeast in some ways. These are cultures that generally work somewhere between mid to low 20 degrees Celsius or 90 freedom units. The end results are generally a much harder cheese that's being pressed considerably more. This is simply because, well, making a cheese at this temperature is generally more viable and is far more common. That is, you really can get away with keeping it in a cabinet away from any possible contamination. This is compared to the thermophilic cultures, and they work somewhere considerably higher, although it might be more accurate to say in a wider range, but they're generally described as being a warm temperature culture. The broad range is anywhere between 20 and roughly 50 degrees Celsius or 68 to 120 freedom units. They're described as heat-loving bacteria, and these are what are used in those much drier, more potently flavoured cheeses. For example, mozzarella, Parmesan, Romano, and more. When you look at this, you may think, okay, there are two cultures, but how do you apply these to cheese? And the way they're used is what is called a starter culture. The starter culture, as the name suggests, is what begins the process of turning the milk into cheese. You will often find that the process for the initial stages is reasonably consistent across all cheese making. That is, you somehow turn the milk into the curds and whey. You remove the whey, leaving only the curds, which are generally fat and protein rich, behind, along with a degree of sugar. That sugar is primarily lactose. To this, you add the starter culture, which would either be the thermophilic or mesophilic culture, appropriate to the exact kind of cheese you're making. The exact breakdown will vary slightly between different kinds of cheese. As mentioned, their first job is to convert the lactose into lactic acid. Lactose is a sugar, and the metabolism of it by yeast creates lactic acid. This has the benefit of adding flavour, but also somewhat inhibiting the growth of other bacteria. This is one of the other reasons why bacteria is used in cheese making. You are fundamentally choosing the least, let's say, undesirable bacteria that also have the benefit of creating a nice flavour. And in doing so, you are preventing pathogenic or particularly dangerous bacteria from growing in the cheese and turning it from something that is edible to something that will give you food poisoning. When you're looking at the actual cheese composition itself, all the species that are used would have to be somewhat tolerant to acidification of it, the redox reactions that occur, the presence of lactate and acetate. This is one reason why lactic acid bacteria are particularly common, although by no means are they the only bacteria you will find. The reason lactic acid bacteria are notable is that they release enzymes that break down milk proteins, primarily casein, into much smaller, initially peptides and then amino acids. These are one of those things that contribute to the umami-like flavor of some cheese types. Once the initial population of bacteria within the cheese has risen to its peak, it then subsequently dies off. But what is important here is not just that it dies off, but then a second generation or subsequent colony will start growing from a different strain of bacteria. As this rises, it makes use of the different elements within the cheese, and these then add complexity and flavor. And you normally have 2 or 3 cycles of this, with different bacteria either using byproducts of the previous bacteria, or in some instances, being able to use products that the first or second generation did not use at all. When we talk about the different kinds of bacteria, it is worth talking particularly about the thermophilic species, largely because those that are able to survive at higher temperatures tend to be, let's say, more problematic. The most common species you'll find within cheeses are your Streptococcus thermophiles, Lactobacillus helveticus, and Lactobacillus delbrueckii. These are both well known to be associated with Central European, largely Swiss and Italian type cheeses. These are also species that, in the case of Streptococcus, can be particularly nasty if they're not kept in check. And this is one of the advantages of having multiple different bacterial strains. The fact that there's intercompetition between them, but also the fact that they're generating products that inhibit the other from getting out of hand. Other notable examples of species that are useful are things like Lactococcus lactis and Leucocontus mentatoides. These are both found in different cheeses again. Perhaps the best way to describe why multiple bacteria are used is not necessarily just to get that complexity of flavor, but to divide and conquer. That is, by mixing multiple different kinds of bacteria together, you prevent any one from taking over and becoming dominant. If no individual bacteria is dominant, it can't become as pathogenic. That is to say that even under the best of circumstances, there is a reason why many cheeses are treated in such a way as to reduce the bacterial population once they're available for consumption, or at least methods are used to keep the population in check. For example, salt. These are meant to avoid issues to do with the bacteria going rogue and causing problems. However, much of that need is reduced by having them compete against each other while still producing desirable flavors, and being able to work successive generations where they use up the material that's available, go away, and a new species rises in their place, only to die off in turn. This means that you get the most possible result out of a relatively limited basic material, that being milk. Thank you for watching this video. If you have found it interesting, consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions you have below.